Okay, so our next lecture is going to be on our single carbon metabolism, single carbon donors. And we did go over these a little bit in the last lecture in order to introduce them so you can understand what was happening when we were making and breaking down different amino acids. And so for this, we're going to focus more on our tetrahydrofolate, vitamin B12, and our S-adenos methionine, or SAM. So these are our single carbon donors. We've talked about biotin, our pyruvate carboxylase, for example, pyruvate carboxylase enzyme used biotin, and biotin, of course, uses carbon sources from either carbon dioxide or bicarbonate. And so it's really limited in where it can get its sources of carbon. Tetrahydrofolate, it comes from folic acid, and tetrahydrofolate has different species, and each of these species have a specific reaction that they participate in, and we're going to talk more about what those reactions are and why they're so important. We also have SAM and vitamin B12. Vitamin B12 has a very important role in formation of SAM, and SAM is, of course, going to be essential for many biologic processes that require a donated carbon, and it's going to be the preferred donor for some reactions in the body. So biotin, again, is going to take carbon dioxide or bicarb, and the reactions are going to be two major ones, which are pyruvate carboxylase and so if this is taking a 3-carbon pyruvate and making it into the 4-carbon oxaloacetate. And this takes place, of course, in the mitochondria. And then there's also acetyl-CoA carboxylase. And this is going to take acetyl-CoA to malonyl-CoA. So these are just two examples. And this is a 2-carbon to a 3-carbon. And so both of these are going to use biotin to transfer those carbons. Now, tetrahydrofolate is going to be made from the vitamin folate, so B9 folate. And it is the primary 1-carbon carrier in the body. The vitamin is able to obtain 1-carbon units from serine, glycine, histidine, formaldehyde, and formate. And then the tetrahydrofolate associated with this carbon can either be oxidized or reduced. So our carbon donors, and we'll talk more about these. So folate exists in various forms, and these are all called the 1-carbon pool. Once the carbon has reduced to the methyl tetrahydrofolate, it cannot be reoxidized. So methyl tetrahydrofolate tends to accumulate inside of cells. Okay, so for the structure of our tetrahydrofolate, we have folate, and this can, again, take on many different chemical forms. The coenzyme form that is going to accept one carbon groups is the tetrahydrofolate polyglutamate form. But this is just simply called tetrahydrofolate. So whenever you see that tetrahydrofolate, it is referring to the polyglutamate form. And so what does that basically mean? So there are three major structural components of the tetrahydrofolate. You have your ring structure, and then you have your para-aminobenzoic acid. That's what that PABA is. Let me write it here. Para. And then you have your glutamate. And those are going to be joined by an amide linkage. So here's the linkage for the glutamate. Now, again, when the term folic acid or folate is applied, it's the most oxidized form of the ring, the pyridine ring. And most of the folate present in the body is present as the reduced coenzyme form of tetrahydrofolate, FH4. Now, the vitamin folate, again, we talked a little bit about how it's absorbed. So, vitamin folate in the diet. And this is going to go into your intestinal cells. And then within the intestinal cells is going to be converted to principally the N5-methyl tetrahydrofolate. And this goes to the portal vein. And then this is, of course, going to be stored in the liver. And then within the liver, tetrahydrofolate gets reconjugated to the polyglutamate form. A large portion of folate will then go into the bile and then gets reabsorbed. And then N5-methyl tetrahydrofolate is going to be the major form of folate in the blood, and this is going to be associated with serum albumin. And that's how it's going to get to move around the body. All right, so the tetrahydrofolate can undergo oxidation and reduction of its one-carbon groups. The one-carbon groups transferred by tetrahydrofolate are either attached to the nitrogen 5. So, and we talked about this in our previous lecture. So the one-carbon groups are attached to our position 5, which is nitrogen 5, or a nitrogen 10, or a bridge between nitrogen 5 and a nitrogen 10. And so, again, these are all going to be referred to as the one carbon pool. The carbon can be oxidized and reduced. The most oxidized carbon is going to be in the N10 formyl tetrahydrofolate, and the most reduced is at N5 that we talked about. So once the methyl group is formed, it cannot readily be oxidized back to N5N10 methylene tetrahydrofolate, and this form will tend to accumulate in the cells. Okay, so now let's talk about our sources of tetrahydrofolates. And this is an image from your book, and it looks very complicated, but it actually is pretty straightforward. So there are carbon sources, and the carbon sources are going to be serine, glycine, histidine, and formate. So we have serine, glycine, histidine, and our formate. And formate is going to come from the breakdown of tryptophan. The donors transfer carbons to folate at different oxygen states. Serine is going to be one of the main carbon sources. Its hydroxymethyl group is transferred to tetrahydrofolate, and this is reversible, as we saw before. So you can go back and forth between serine and glycine. And it's catalyzed by serine hydroxymethyl transferase. Right, and then you form that N5N10 methylene tetrahydrofolate. Now you also have recipients of the one carbon groups. Okay, so transfers are going to be involved in the synthesis of glycine from serine and the synthesis of the base thymine, which is required for DNA synthesis. Both the purine base is required for DNA and RNA synthesis, and the transfer of methyl groups to vitamin B12. So we have here our purine biosynthesis, which is going to require the N10 formyl tetrahydrofolate. We have our transfer to our vitamin B12 to generate SAM, which is going to be a methyl donor, and so that is going to be by the N5 methyl tetrahydrofolate. And remember, we said that the N5 methyl tetrahydrofolate is difficult to reoxidize, but this is one way that you can get transfer of that carbon, and it regenerates tetrahydrofolate. You also have the deoxythymidine monophosphate, DTMP, which is produced by deoxyuridine monophosphate, DUMP. And this is a reaction in which the DUMP gets methylated to form the DTMP. And the two hydrogen atoms from tetrahydrofolate are used to reduce the donated carbon to the methyl level. And so this is going to use that N5N10, methylene tetrahydrofolate, and you are going to regenerate tetrahydrofolate, which requires NADPH. Okay, so the recipients, let me get a different group, are going to be numbered in this particular diagram, numbers 5 through 8. So here's 5, which is our purine. Here's 6, which is our TMP. Our 7 is going to be our N5 methylene tetrahydrofolate, and our 8 is our vitamin B12. Okay, so now we talked about, we had this picture in our previous lecture about the different forms of tetrahydrofolate. And of course, our tetrahydrofolate, remember, are 5, 6, 7, 8, 9, and 10 is numbered. And so tetrahydrofolate has a hydrogen group on 5, 6, 7, and 8. And that's the 4 hydrogens that give it this tetrahydrofolate. N5 methyl tetrahydrofolate, which is the most reduced. And it's also important to transfer to vitamin B12. And this again eventually will form SAM. We have the N5-formyninone tetrahydrofolate, and this is part of histidine degradation. So if you go back just for a moment to this slide, you can see that we have histidine, different color, and it has an N5-formyninone tetrahydrofolate as its in its pathway. Eventually, histidine will generate the N5-N10 methyl tetrahydrofolate. We have the N10-formynyl tetrahydrofolate, N5-N10, which is involved in a lot of donations, and we have the methylene and the methanol, which are those bridges. Okay, so our one carbon pool overview is just that carbon is transferred by biotin if it's in the form of carbon dioxide or bicarbonate. Other one carbons that are less oxidized are going to be transferred by reactions that involve tetrahydrofolate, vitamin B12, and S-adenosylmethionine, or SAM. And so this is just showing you our carbon donors up here are serine, glycine, histidine, formaldehyde, and formate, which, and then we have our different forms of tetrahydrofolate that will come from this. We have our tetrahydrofolate, which will be picking up those carbons, and so this tetrahydrofolate can go in between different forms of the one carbon pool, and all of these different forms are going to lead to products of the after receiving the carbons from these one carbon pools. And so we can form purines, we can transfer carbon to vitamin B12, which will then be important in producing SAM, as well as DTMP from UMP. So just a side about folate deficiency, we've talked about this already. You can lead to megablastic anemia, which you have very large, almost ovalish red blood cells. Fewer of them, they get removed much faster. It's similar to a vitamin B12 deficiency. And this is caused by an inability of your precursors to synthesize DNA. So as you saw for folate, tetrahydrofolate was very important in purine formation, for example. And so if you're not able to make your purines, you're not able to synthesize DNA, and cells that need DNA, rapidly dividing cells, such as red blood cells, the formation of them will be impacted. Folate deficiencies also, you have neural tube defects, which we've talked about, spina bifida, and so mothers typically will take folate as a precaution, or pregnant women will take folate as a precaution. Okay, next are vitamin B12. So B12 is known as cobalamin. It contains a corrin ring, and this, as I said before, is similar to the porphyrin ring found in heme. Difference between them are 2 of the 4 chiral rings in our vitamin B12 are joined directly rather than by a methylene bridge. It also has the unusual feature of cobalt being present, and cobalt can form a bond with carbon. In the body, it's going to react with the carbon of the methyl group, which is going to form methylcobalbumin. It can also interact with the 5' carbon of 5' deoxyadenosine, and that forms a 5' deoxyadenyl cobalbum. So X, let's see, if X equals your 5', and this would be in the deoxy. Put in 1 here. 2X could be our methyl, and this would be Or 3, X could be our cyano group. And this, as I said before, is going to be the major commercial form. Or in drugs, for example. Now, vitamin B12 absorption is very specific. So most of the sources of vitamin B12 are going to be in meats, eggs, and dairy products, also in fish and poultry and seafood. The animals that serve as a source of the vitamin B12 is coming mainly from the bacteria in the animal's food supply. So it's actually bacteria that make B12, and then the animals feed contain bacteria plus the B12, and then humans eat the animals plus the B12. That's pretty much how it goes. Absorption is going to be very complex. Vitamin B12 can be either free or bound to proteins. And if it's free, vitamin B12 binds to proteins known as R binders. So here we have dietary B12, and this is showing that it's free. And so it's going to bind to what's called an R binder. R binders are proteins that are released by your gastric mucosa cells. And the R binders are haptocorins. And these will be released not only by the gastric mucosa, but also by salivary glands. Now, if the ingested B12 is bound to proteins, it has to be released by digestive proteases first. So in the small intestines, proteases will digest haptocorins, and the B12 binds to them the intrinsic factor. So your dietary B12 will bind to R binders or be bound to proteins. These will be digested by enzymes, release your B12, and then the B12 is going to bind to that very important intrinsic factor. Intrinsic factors are released by your parietal cells, and your intrinsic factor bound to your vitamin B12 can then bind to your transcoalbumin. And so this is the receptor for this. The B12 within the enterocyte will complex with the transcoalbumin and then release into the circulation. The liver will take up about 50% of this, and then the liver will store this. Storage of vitamin B12 can be for up to 3 to 6 years. So if you have a deficiency, you're not going to really present with it until about 3 to 6 years after you're unable to, for example, absorb vitamin B12. Now, a vitamin B12 deficiency is going to be mostly by a lack of intrinsic factor, and this is typically due to a autoimmune disease, as I've mentioned before. Now, one of the consequences is going to be the accumulation of that methyl tetrahydrofolate. As I said before, the most reduced form of tetrahydrofolate cannot easily be oxidized. And so one of the big ways to get rid of this form is to transfer that methyl group to vitamin B12, and then you can regenerate tetrahydrofolate. And so if you have a vitamin B12 deficiency, if you cannot go, if this does not happen, maybe I should, hang on. Then this is known as the methyl trap. And so basically you cannot get rid of those carbons that accumulate in the cells in this most reduced form. And this is because you cannot transfer them to vitamin B12. And this will lead to a functional folate deficiency because all your folate eventually get trapped into that reduced form. Okay, so now let's talk about some functions of vitamin B12. There are two very important reactions that the body uses vitamin B12 for: the transfer of the methyl group from N5-methyl tetrahydrofolate to homocysteine. And so we have here homocysteine, and you have this B12 with that methyl group from N5. So back here you would have N5. And this would regenerate tetrahydrofolate, transfer that methyl group, and then you can go on to form methionine. And also, you also have the second one, if you guys remember, the transfer of your B12 is important for your odd-chain fatty acids and going from that step of methmalonyl-CoA to succinyl-CoA. Now SAM is going to be downstream of your methionine and that whole transfer of your methylene group from that N5-methyl tetrahydrofolate to vitamin B12, which was up here, to our homocysteine to create methionine. And then from methionine, you can create SAM using ATP. And so those are the two precursors. And it's going to be produced when the your adenosine triphosphate, your adenosine portion gets attached to your methionine and your triphosphate gets released. And so you have your ribose with your adenosine here, and this forms S-adenosyl methionine, or SAM. And SAM is going to transfer the methyl group to precursors in a number of compounds. So SAM transfers carbon 2, for example, in formation of creatine, phosphatidylcholine, remember that story? Epinephrine, melatonin, methylated nucleotides, methylated DNA. And remember, we talked about methylation of your DNA and how it's important for silencing genes that are not necessary to turn on. And so SAM has an important role in making sure that those are properly methylated. So SAM is going to be very dependent on tetrahydrofolate and vitamin B12. Keep in mind that homocysteine is derived from methionine metabolism, but can be reconverted back to methionine with both the methyl tetrahydrofolate, the most reduced form, transferring that carbon to vitamin B12. And this reaction is the only reaction which you can use that most reduced form. So again, you have your, I guess I'll do it here. And then this with homocysteine, and you regenerate tetrahydrofolate. The key here is being able to go from here to there. And so this is the only reaction that allows you to do that. Another thing to keep in mind is that if you do have a vitamin B12 deficiency, you're going to have elevated levels of homocysteine. And so elevated levels of homocysteine have been linked to cardiovascular disease as well as neurologic diseases. So not only if you have a vitamin B12 deficiency are you going to have trouble making SAM, you're also going to have a buildup of homocysteine, and that in itself can lead to problems as well. So SAM is going to be used in reactions that add methyl groups, and these methyl groups are going to be added to either oxygen or nitrogen atoms. So we'll add that here. And this is different than folate derivatives, and folate derivatives are going to add their one carbon groups to sulfur or another carbon. So this is the difference between SAM and tetrahydrofolates, for example, the different versions of it. So there are many reactions that require the methyl donation from SAM, and SAM is going to transfer its methyl group and then be rehydrolyzed to form homocysteine and adenosine. And so homocysteine is going to be coming from the methionine that is essential in the diet. And so the homocysteine portion of methionine is the essential portion. However, there's no good dietary source of homocysteine, whereas methionine is plentiful in the diet, so that's why methionine is essential. Now, the relationships between folate, vitamin B12, and SAM at this point hopefully is very clear. There is the methyl trap hypothesis. So within the folate cycle, the equilibrium lies in the direction of N5-methyl tetrahydrofolate. Let me change this. And this, of course, is the most reduced, as I've said. It cannot be readily oxidized, and it has only one option to donate that methyl group to vitamin B12. And so the reason for this is because it's the most stable form. Of the tetrahydrofolates. The methionine synthase reaction is going to require vitamin B12, but the only way for vitamin B12 to get a methyl group to transfer to homocysteine to form methionine is from your N5-methyl tetrahydrofolate. Okay, so these are all interconnected. You have here your carbon that's going to be transferred to your vitamin B12. You're going to regenerate tetrahydrofolate. You're going to add that carbon to B12. B12 will donate that carbon to homocysteine, and then you're going to generate methionine. All of this is very important because ultimately you're trying to get your SAM produced, and then SAM can transfer its carbons to many precursors to give you some product plus a carbon. So the methyl trap hypothesis is basically if vitamin B12 is deficient or if methionine synthase is defective. Let me write this a little bit better. So the methyl trap hypothesis. So if B12 deficient or methionine synthase is defective, then your N5 accumulates. It gets stuck, and that's considered the trap. And so most of your folate will get trapped. So folate derivatives will all go towards here, and then they get trapped. So a functional folate deficiency, or the appearance of a functional folate deficiency, may be caused by a lack of vitamin B12 or a deficiency of the methionine synthase enzyme. And so any of this, whether it's a folate deficiency, vitamin B12 deficiency, or lack of function of the enzyme, is the methyl trap. Now there is also the hyperhomocysteinemia, and so again, that's elevated levels of homocysteine have been linked to cardiovascular and neurologic diseases. So methionine is always going to be in our diet, and methionine is going to form SAM, and then it's going to generate homocysteine. And if there is, again, a vitamin B12 deficiency, or if there is a defect in that synthase, you can lead to an increase of homocysteine. Homocysteine is going to, of course, be the byproduct of SAM. After SAM donates its methyl group, SAM will become homocysteine. And homocysteine produced can either be remethylated to methionine or condensed with serine to form cystathione. And we talked about that in the previous lecture. So if an individual is deficient in vitamin B12, then the conversion of homocysteine to methionine, which is the major route, gets inhibited. And as cysteine accumulates, this pathway also gets inhibited because cysteine is a negative regulator of its own formation. So overall, you're going to lead to an increase of homocysteine if you have a B12 deficiency, for example. And then your homocysteine is going to get released into the blood. Now, hyperhomocysteinemia is also going to lead to an increase of homocysteine if a mutation is present in the enzyme that converts N5-N10 methylene tetrahydrofolate to N5-methyl tetrahydrofolate. So go back to this picture here. We have our N5-N10 methylene tetrahydrofolate, which has to become N5-methyl tetrahydrofolate. And this is by a N5-N10 methylene tetrahydrofolate reductase, which requires NADH. And so this is taking your one carbon donor back to the most reduced. And then this, of course, will donate to vitamin B12. And then this will be B12 with your carbon. And you have your tetrahydrofolate, which will get regenerated and can go back to being tetrahydrofolate, pick up other things. B12 with homocysteine makes methionine. And this is a major route. So let me put some numbers here. So 1, B12 deficiency. 2, synthase defect. And 3, an inability to form your N5-methyl tetrahydrofolate from your N5-N10 methylene tetrahydrofolate because of a defect of the reductase. And all of these will lead to hyperhomocysteinemia. Okay, so there's many ways to get hyperhomocysteinemia. Now, there also are folate deficiencies. So just very briefly, we have, again, those neural tube defects. Folate deficiencies are also going to lead to an increase in uracil incorporating into DNA. Now, uracil can usually be removed by DNA repair enzymes, but the lack of that DUMP to DTTP is going to block this. So this results in fragmentation of the DNA and a block to normal DNA replication. So you're going to see with patients with this, you're going to be able to isolate neutrophils, which are cells of the immune system, and they're going to have clumping and polysegmentation in their nuclei. And so this is a sign of megalastic anemia, primarily caused by folate deficiencies or a vitamin B12 deficiency. Oops. And so with folate deficiencies, the damage to cells that are affected is not repairable, and the cells will undergo apoptosis. Now there is also talk a little bit about choline, because choline is going to be essential for some of our phospholipids, and choline is oxidized to betadine, or betaine, sorry, betadine, betaine. Choline gets oxidized to become betaine, and betaine can be used in the liver to donate a methyl group to homocysteine to form methionine. And so this is sort of a way that the liver can be able to make SAM in the absence of vitamin B12, for example, so we can get around that whole process. But this can only be done in the liver. So in liver, choline gets oxidized to betaine, and betaine can donate a carbon to homocysteine to make methionine. Therefore, the liver has two routes for homocysteine conversion, whereas our nervous system or any other system only has one route, the vitamin B12 pathway. Okay, and so that is this end of our one carbon donor lecture.